Hello, everybody. My name is David Leclerc. I'm the host of the ChUG podcast. We are today talking about autofluorescence in spectral flow cytometry. Our guests are Monica Delay, who is the US Manager for Application Support with Cytek Biosciences, and Laura Johnson, Laura Johnson, who is the here at the University of Chicago. She's the Associate Scientific Director in the CAT facility. Hello, and thanks for your time, Monica. Thanks for joining us.
No worries. This is fun. It's always good to catch up with you guys.
Yeah. The reason we wanted to talk about autofluorescence today, we're just a few weeks past CYTO 2021. And again, spectral flow was discussed with much gusto. And within those discussions came the topic of autofluorescence, which people has started to identify as sometimes a problem, sometimes possibly something that can be used to study cells. And we essentially wanted to get a rough idea of what to expect when you think about autofluorescence. I think the main goal I have for the listeners of this podcast is if anybody start talking about autofluorescence, the listener of our podcast will be able to look at that person straight in the face and say, I know what you're talking about. That's the general goal.
Sounds good.
So Laura has been working on multiple projects at the University of Chicago and has been in touch, it's been dealing with quite a few problematic signatures that we got on our Cytek Aurora. And Monica, as in your position, you are, exposed to all kinds of crazy piece of data, and so I think it's a good match here. It's a good discussion we're going to get. Maybe before we get started, so you work for Cytek, but it's something we told at the last podcast. The ChUG podcast is not supported by industry or by the University of Chicago or my parents. It's not supported by anybody. And so we can, and secondly, we're going to talk about the data we're going to be talking about is coming from Aurora's instruments. And maybe we can clear that out of the way. I'm not sure what we would expect from other companies that produce spectral flow instruments. So we're talking about Sony, Thermo Fisher as the Bigfoot now that also can generate spectral flow data. Do you have any idea of what kind of difference? I think the Bigfoot, I'm not even sure if they do autofluorescence subtraction, if they even look at it yet.
That's a good question. Yeah, I don't know. I'm not familiar with their software. I have seen Sony speak about their tool. I think it's called like an autofluorescence picker, maybe. But I think at CYTO Virtual, there was a Sony rep in one of the spectral breakout sessions, and he talked a little bit more about it. Yeah, so I'm not familiar with their software, but certainly you can select multiple autofluorescent signatures, like we've worked with Laura to do with some of her data sets, and then she's kind of taken off with it for some of your users. Yeah, but certainly, I mean, really, even when you're not using full spectrum cytometry, if you're just using conventional cytometry, you could select a channel to assign to autofluorescence, which is certainly what we used to do. And when I was looking at references for this podcast, it's very interesting because as I mentioned to you, it's like the pioneers in flow cytometry that we've been, listening to at various meetings, give talks. And I just happened to find a publication from 1986 that Mario Roeder and Bob Murphy were on. And then from 1987, Dave Parks and Len Herzenberg, Leonard Herzenberg were on. And they kind of used the same approach, except for me, I think it was different wavelengths. that they were using to kind of assign a channel autofluorescence so that they could subtract it out of their data and get better resolution. So, back in the late 80s, people were already, kind of trying to figure out how to deal with it. But of course, that's evolved now that we're looking at so many more channels. Yeah, I would imagine that with the Bigfoot, you would use the same approach. You just have to turn on all the channels, or as many as you feel are necessary to give you a good signature. So I forget how many channels I have.
On the Bigfoot?
Mm-hmm.
54, I believe. Might be wrong.
Okay.
Yeah.
Roughly.
Something in the 50s.
Yeah.
Mm-hmm. Mm-hmm.
Yeah, I think the general concepts we're going to talk about today can be broadly applied to other technologies or even a conventional flow cytometer, although we'll kind of mention some of the specifics for how you actually set it up on the Aurora, but we're going to try to stay away from that a little bit.
Sounds good.
So maybe that's the first question, just to start the discussion. And Laura and Monica has prepared quite a bit for this discussion, and I have not. So that will become quite apparent pretty soon. So when we first started here at the University of Chicago with our spectral flow instruments, we were just running things without thinking too much about the autofluorescence of the cells. And then soon enough, some project involving either cancer cells or lung tissue came up, and suddenly, oh my God, these things are problematic. It's really hard to unmix. It's really hard. It's causing all kinds of issues. And that's where we got started with discussions with autofluorescence. So I guess my first question is, like, where do you start? Where do you have a point where you don't think about autofluorescence, and at some point you go, oh wait, I'll need to do something about these really, really bright cells?
Or do we want to back up a second and define exactly what autofluorescence is? Just
yeah, sure.
Make sure we're all on the same page. So basically, if you take cells, any cells, mammalian cells, bacterial cells, plant cells, and you don't add any additional fluorophores to them, you just run them unstained on a flow cytometer, then many cells will have a fluorescent signal on their own. It will be relatively low, typically, in comparison to a fluorescent signal. But with the spectral cytometers, we can actually see a somewhat unique looking signature that can be found in just any unstained cells. And so that's what we're talking about when we say autofluorescence, is our unstained spectral signature. And that can change based on the cell type or a bunch of different things. We'd kind of list it out, but in general, it comes from things inside the cell. Different molecules inside the cell can lead to different autofluorescent patterns, and there are different processes that affect autofluorescence, like the metabolic state, or I'm assuming like the stage of cell growth. And there's like a whole host of things that can affect exactly what this autofluorescence is. But the basics of it is unstained cells.
Well, it's interesting, if you go back to what I was saying before, is like in conventional flow, when we've had to deal with autofluorescence, we've just assigned a channel to it. But, the more channels you have, and, conventional flow, you could view the data the way that we view it in spectral. Like, you would just have to set up the software display differently so that you could take all of the data from the channels and view a spectral plot, even though it's not a full spectrum cytometer. So I just think we haven't really looked at that before, right? So we haven't really explored what an actual signature looks like in I guess you could call it a lower resolution cytometer versus a higher resolution cytometer where you're sampling, wavelengths in a more narrow range. So I keep, it's funny because we keep thinking about, it comes down to almost just display, right? So between the two types of technologies, and really understanding, what cells look like by themselves. It just gives us much more resolution when we have many, many more channels, and we're able to really see the signature in the way that we display the data.
All right. So back to my question then. So when do you start worrying about autofluorescence? I guess the general idea with traditional flow, whenever we talk about it, was we just get brighter signals to separate the background noise or the autofluorescence of the cells from the actual signals coming from the fluorophores. And so it's just a push forward. With when you look at autofluorescence, I guess you're looking to like reduce that influence and reduce the noise ratio, the noise signal ratio, I guess.
Yeah. I think it's, to me, I mean, you kind of hit it, is when the resolution is compromised for whatever marker you're looking at. And, I think it's, I kind of was thinking about, when is autofluorescence an obstacle to overcome versus a tool that you can use, right? And so, I think it depends on the assay, and it depends on the, tissue type and how many markers you're looking at. But certainly if your resolution is compromised because you have high background, that's when it's problematic, I guess, would be my view on it. I don't know if Laura, from a user perspective, yeah.
I've started to add to that. So I kind of have divided autofluorescence into a bunch of different categories or scenarios. I don't know. So the first division is in thinking about how the software on the Aurora utilizes the unstained control, right? Because the autofluorescence is in the unstained control. So anytime we use that, we have to factor in autofluorescence. So there's two cases in the software where the unstained control is used. One is in the reference controls when you're setting up the unmixing. And so autofluorescence is a component of setting up the unmixing algorithm. And
you're talking the single stain controls.
The single stain controls. So it can be factored into that component of your experiment, or more what Monica was talking about when we're looking at our fully stained samples and our different tissue types, and trying to figure out the best resolution of our markers. It can also be factored into that as well. So I can actually pull up some slides. So I guess if you're listening to the audio, then you won't be able to see this. We'll try to explain it. But if you're on YouTube, we have a couple visual aids. So as far as the reference controls, it's particularly important to pay attention to the autofluorescence if you are in situations like what I'm showing for these two single stain controls. So one example is you're staining CD14 on whole blood. So CD14 positive cells are mostly going to be monocytes, and your CD14 negative cells in that tube are mostly going to be lymphocytes and granulocytes. And those different subsets of cells have different autofluorescence properties. And so in order to get the unmixing algorithm to work as best as possible, you want your CD14 positive population and your CD14 negative population to have matching autofluorescence properties. And so if we use this tube and use what I call the internal negative, so the CD14 negative cells within the CD14 single stain tube, then those CD14 negative cells, autofluorescence does not match the positive cells. So the algorithm isn't going to work quite as well. And so instead, what you would want to do is use a universal negative. So it would tell the software, instead of using the CD14 negative cells in the CD14 single stain control, use an unstained tube. And then that's where we would want to gate on monocytes so that we would have a matching autofluorescence population with the CD14 positive cells. Or another situation is, for example, if you're staining CD45 and pretty much all of your cells in the blood are CD45 positive, and you basically just don't have a negative population at all in that tube, or it's very, very small, then that would be another situation where you might want to utilize the universal negative, and you have to make sure that the autofluorescence properties match. So depending on how complicated your experiment is, if you have multiple tissues in your experiment, you also have to make sure that you're matching up the right unstained tube with the right stained tube in terms of autofluorescence properties. So there's that situation for the controls. And then for the fully stained samples, this is the next slide, yes. I think of autofluorescence in 3 different categories. So the first category would be simple autofluorescence, and it would also be a low signal. So the examples we're showing are human PBMCs or mouse spleen, and you can see that overall in these tissues, there's just a single autofluorescent signature, and it is a relatively low or dim signal. It's category 1. And category 2 is still simple autofluorescence. So there's still just one signal in the overall tissue, but it's relatively bright. So that is a separate category. So I'm showing epithelial cells. I'm not entirely sure about this specific sample. I don't know if we can broadly categorize that to all epithelial cells, but this sample that I happened to get fell into this situation.
Are those like ex vivo, or is that a cell line?
It was a human biopsy sample.
Oh, okay. Yeah.
Yeah. And then the third category is complex autofluorescence. And when you run that, so the example we're showing is whole blood, you can see that there is more than one autofluorescent signature within the entire tissue, and we could actually divide those out. So I'm showing the signature of the neutrophils separate from the signature of the lymphocytes, and those each have their own signature, but the tissue as a whole has multiple signatures. Or I have another complex example here where this is an unstained tissue digest, and I'm showing 4 different signatures, but there may have even been more than that. So those are the 3 categories I would call tissues: either simple but low, simple and high autofluorescence, or complex autofluorescence.
All right. So I want to tread back on that just to be sure I'm following. So the first case is just a matter, we're back to the rules of your unmixing controls, right? You just want to make sure, you know, your control is as brighter than what you'll find in your sample, and one of them is also match your the autofluorescence of your positive and negative fractions. So that's the first case. The second case.
One thing I wanted to add there, that's true for comp beads too, right? So if you're using compensation beads as your single stain control, your negative population has to be just the autofluorescence of the unstained beads, so that you're properly, I mean, you can think about it as normalizing the signature. So you're subtracting out the background, so you understand what fluorescence is only coming from the fluorophore.
And in this case, you would actually subtract the autofluorescence of the cells to run, or would you just gate specifically on the same type of cells and use that as a control?
Oh, the algorithm subtracting the background.
Okay.
Yeah. So you don't have to do it yourself, but you have to understand what is just the background of that particle. And so, you know, some of the common things we see in, for example, people using compensation beads or even sometimes cells, as they don't wash properly. And so then there's residual fluorophore signature on the negative population, which will incorrectly subtract the background. And it's so hard to convey this when we're teaching sometimes, because, you know, you have an unstained and then you have a negative, and people get confused about what each one is for, which is why it's important that Laura is going through this explanation. So,
yeah, the way I typically explain it with my training slides is that the algorithm is going to assume that within your positive population, there are two signatures, the signature of the fluorophore and the signature of the autofluorescence.
Of the particle, yep,
You
exactly.
assume that the negative population contains only the autofluorescent signature. So for the algorithm to figure out what is the fluorophore signature only, it's going to take the positive population and subtract the negative population from it. So it's
Correct.
the autofluorescence. So if your negative population does not match the autofluorescence of the positive, then the math gets thrown off.
Sounds good.
So, and it's funny because there are some, I won't say what software programs, but there are some software programs out there that just use a universal negative for the compensation matrix. So then you have your unstained sample. And if you're using comp beads for your single stain controls, it's using the unstained sample as the universal negative, which is incorrect.
Yeah. And it can even get more complicated if you have markers on different subsets. So you have some neutrophil markers in your panel and some lymphocyte markers in your panel, and you use the universal negative, but sometimes you want that gate to be on neutrophils and sometimes you want it to be on lymphocytes. So I have a whole video explaining all of this in detail with a tutorial. We could put the link to that on.
That'd be great. Yeah.
That's a good idea. Follow that tutorial. We won't go into the
For
details.
those listening on the audio, you probably will need to find the YouTube version of this podcast, and we'll have all the links below in the little text box below. All right, so that's the first case. The second one refers to what Monica was discussing before. So just a case where you have such a high level of autofluorescence background coming from the cells themselves that it makes it difficult to resolve your actual positive signals. And that will be specific to specific cells. Like in this case, we have epithelial cells, which have pretty high background. I think it's 10 to the second, 10 to the
I think
third,
the grade
the fourth.
is 10 to 5.
Oh yeah. So
yeah, those are pretty high. Those epithelial cells.
They're pretty extreme.
Right. And so, all right. So the solution here is going to be to do what is called subtracting the autofluorescence. And I don't think we've discussed what exactly that entails yet. So maybe we should dig into that.
The autofluorescence extraction.
Yes.
Yeah. Yeah, there are a couple ways to do it. And I think this goes back to you were asking, when do you use autofluorescence extraction and when do you not? And then when do you go to further and not use just our autofluorescence extraction tool, but when do you actually define it as a floor in our library? And this is kind of specific to the way that our software is set up. I don't know how other companies do it, but so these are kind of my rough guidelines would be, I feel like once the autofluorescence, like the highest channel that you have autofluorescence in, once it hits about 10 to the third, that's when I think, okay, we could we'll benefit from autofluorescence extraction. Then I feel like once that highest channel of autofluorescence hits 10 to the fifth, I would say, and maybe slightly less, that's when I would probably define it as another floor, like an extra floor. And then I try to find a negative population to use the universal negative if it's the same cell type. It's very rough. I don't know if my colleagues that work for Cytek would agree with me, but that's like kind of my experience in working with different autofluorescent tissues or samples.
Is there a downside of dealing with autofluorescence with pretty much any samples that you look at? Like say you look at your lymphocytes and the graph you were looking at have a pretty low amount of autofluorescence. If you try to remove the autofluorescence from these cells, is there a downside to it, or?
Yeah, sometimes it'll overcorrect, and then you might get some slight negatives, like off of some of the populations. We've seen a little bit of that using the UV laser. After we added the UV laser to our system. And I'm not sure why that would be, but it could be just the way it's taking the negative population, like how it's defining the unstained, and so I'm not sure. I'm not sure. I haven't really like explored that a ton. It's because I'm always just trying to get to the end where my unmixing looks good, right? So it's like kind of tweaking where I'm putting gates or kind of the way that the method that I'm using or the strategy that I'm using until I get some nice looking data.
So you said you just that showed up when you added the UV laser. It was not the case before, or just
I'm sure there would be a way. Like it depends on how you're gating on the population in the unmixing wizard. Yeah. But
no,
I think we saw it a little bit more. We saw it a little bit more with the UV laser. Yeah.
Got it. Okay.
Yeah. Yep.
Hmm.
So yeah, if you select a relatively low autofluorescent population like lymphocytes and PBMCs, and you do autofluorescence extraction, sometimes it might overcorrect. It might think, there's more there than there should be. So, and then you get a kind of an extreme negative off to the left of some of the populations. But it depends on what fluors you have in your panel. We have a slide that shows that as an example. I'm sure we've had it in a webinar somewhere. I'd have to dig it up. So,
yeah, for judging when you have to use autofluorescence extraction or not, I think Monica's guidelines sound good to me. I don't know if I necessarily have done that in my experience. It's kind of difficult to define. So I guess as far as thinking about autofluorescence in the reference controls, everyone should always be considering that. But as far as if you should use the autofluorescence extraction tool or not, it's something that I encourage everyone to play with. I don't know if everyone does it, but like Monica said, your end goal is good unmixing. I guess actually I think there's two end goals. So one is correct unmixing, and the second one is the best possible resolution of your markers. Right. So I have, I guess, I always get the really hard cases coming to me. So I see all the really challenging, very high autofluorescence. And when you have a very bright autofluorescence signature in your sample, I've found that it's really hard to get the unmixing to look correct. So if you're checking unmixing using N by N plots of your single stain controls, it's hard to get those to look good if you have a really high autofluorescence signature until you start to add in that autofluorescence extraction. Then that basically cleans up the N by N plots, and then you can assess your fluorophore unmixing much better. And without the autofluorescence extraction, those N by N plots just look like a total mess. But if you have the simpler cases with the dimmer autofluorescence, then you should be able to check your unmixing and get those N by N plots and everything looks great. But then you might choose to play around with the autofluorescence extraction after that to see if adding in the autofluorescence extraction feature increases the resolution of your markers, or if you get these weird negative effects that Monica was talking about. So it might help and might not. But it's something you could play around with in the simpler, dimmer cases, but in the more complex cases, and when you have a lot of really bright signal, then I have not found that you can successfully make it past the unmixing check.
Yeah, and so, it's funny. So thinking about unmixing errors, which really are the same thing as a compensation error. So it all has to do with, are your controls accurate? The algorithm, all it's trying to do is figure out where your photons are coming from, right? And so, when we go from the raw full spectrum data to the parametric data, it's trying to understand, is this fluorescent signature from your cell itself, the autofluorescence, or is this fluorescent signature from BB510 or, APC or whatever? And so it's really all about assigning photons. And so when you have confusion in your signatures because your autofluorescence looks a lot like BB510, and it's super high, then it could misappropriate it. And so that's where we get the unmixing errors. And so, I just want people to understand that the math isn't magic. It's very agnostic and unbiased. And I like to say that it's an input, there's an input and an output. And if your input is wrong, then your output is going to be wrong. And so, just trying to make people understand that it doesn't have to do with the unmixing algorithm. It has to do with just the complexity of the situation.
Yes, that was going to be the question that was building in my mind. The autofluorescence on top of very complex panels, which results in dealing with all this spectral overlap, will generate this data that starts spreading out. So I guess autofluorescence. So we're trying to decide whether or not to use autofluorescence, and I guess if you had high autofluorescence but still good resolution, be good without it. But as you add more and more markers, eventually the whole bag of problems that gets generated by all this stuff mixed together, very precise vocabulary here, just makes you kind of, this is kind of a tool that you have in your toolbox that just like,
yeah, absolutely,
going forward.
Yeah.
So not to see a question, I guess, but
no, but that brings up another point. In general, the autofluorescent signature, and especially the one that Laura showed, even the simple autofluorescence, it looks kind of like BB510. The signature looks kind of like BB510. And I've seen autofluorescence that has that same signature, but super, super high. And I don't know. And I think it was at about 10 to the 5th I think the channel is violet 7, is like kind of the peak channel for autofluorescence. And
people know violet 7 is about 600-ish.
It's 5. Yeah, I can't remember. I'd have to look it up. I don't have those memorized. Which maybe I should.
No, yeah, that's going to be the sign of true expertise. One day we'll know exactly each of these.
The central wavelength for V7 is. Anyway, so I've had a data set where the autofluorescence was like at that 10 to the 5th mark, and they were trying to use, I think it was, is it live dead yellow or live dead aqua that peaks kind of in that, or it looks really the same as BB510 or autofluorescence. It's about the same signature. And they were, we just couldn't get it to work because the autofluorescence was too high. And so, suggesting a modification of their panel to a different viability stain was really needed. And so, it kind of goes back to how bright is the autofluorescence, how high is it? And we need to make those choices or changes to our panel based on that. And then, you can use that information. You could say, well, okay, so I'm just going to assign this to my autofluorescent signature, and now it's going to become a parameter in my unmixed data. So if I want to look at that against another marker, I can. But in general, we usually don't. We usually just care about our markers and not whatever autofluorescence signature we've assigned.
Yeah, I actually pulled a few of the fluorophore signatures that I thought by eyeball looked like autofluorescence. So I don't have personal experience with every single one of these fluorophores, but I just kind of went through and pulled signatures that I thought looked kind of similar to autofluorescence. So what I was looking for was sort of a overarching signal across the UV laser that peaks in the middle of the UV laser, as well as another overarching signal that peaks in the middle of the violet laser. So what I came up with was the live dead blue, or I guess zombie UV would also fall into that category.
Yeah.
UV 496 looked kind of like that. BV 510 and the aqua viability dyes. Zombie yellow, live dead yellow, pack orange, and BV 480. So yeah, in my personal experience, I have definitely had trouble with BV 510, and I think I had a panel that had zombie UV, which is similar to live dead blue, but it's not quite the same. And so yeah, I had a lot of trouble with very highly autofluorescent cells. These were actually kind of on the extreme case of autofluorescence, but they're very high autofluorescence in the UV. And then using the UV viability dye, it was very hard to figure out which cells were dead and which cells were alive. And also figuring out the best titration for that dye was really tricky. And then I was that depending on how I set gates or which autofluorescent signatures I put into the unmixing algorithm, sometimes the cells would be categorized as autofluorescence, or sometimes they would be put into the viability dye channel. They're kind of like bouncing around depending on how I set things up. So I have definitely struggled with that viability dye and BV510. I don't have personal experience with BUV496 or BV480, but I think by eyeball, I could see how those ones could cause issues. So
I think with BUV496 and BV480, what I notice about those signatures is, well, with BUV496, you have some signal in the first 3 channels off the UV laser, which is not present in autofluorescence. And also you have the signal off of the blue laser, which usually isn't present in autofluorescence either. So to those 2 kind of differences will help us distinguish, right? Because, you know, we're looking at all 64 channels if it's a 5 laser system. And so, you know, any differences between autofluorescence and the floor is going to help us. Whereas I see like live dead blue and BV510 don't really have those other distinguishing features about them. And they do literally almost look exactly like autofluorescence. So especially BV510. Yeah, with BV480, again, you have that pretty major signature off the first 3 channels off of the blue laser, which again helps to distinguish it. So I don't tend to see as much issue with BV480 for autofluorescence, but you know, I suppose if it's high enough, it could be something to consider. But yeah, definitely zombie yellow and live dead aqua are really not my favorite viability dyes for high autofluorescent cells. Yep.
I think it just depends on what your autofluorescent signature looks like.
Yeah, absolutely.
If you have that, you know, simple case like the PBMCs or the mouse spleen, then you should be fine with.
Yep, absolutely.
And but if you have the more extreme cases, which are very, very bright and very complex, then I guess that brings us to one of our talking points was how do we deal with the autofluorescence? What strategies can we use? So strategy we've kind of been alluding to is planning your panel around the autofluorescence that is present in your sample. So you would have to figure out what kind of autofluorescent category you're in with your tissue type. Do you have very bright, complex autofluorescence, or you just have a very simple case? And if you do have a lot of autofluorescence, then figuring out which fluorophores are similar to the autofluorescence patterns that you have, and then not putting those fluorophores in your panel.
Yep.
The rule of thumb used to be you're going to get higher autofluorescence at lower wavelengths. I don't know if that holds true in your experience, or
for the most part, yeah. I feel like for the most part, it seems like the autofluorescence is the highest off the violet laser for the most part. Certainly you get, quite a bit of signal off the UV, but yeah, in my experience, it's mainly the violet. So I was trying to remember what, so the workflow that, I think it was both Alexis Gonzalez who did a webinar for us, and you can access it on our website. And then also Diana Bonilla, she did that webinar for autofluorescence. Well, she did one of the talks for our virtual user group meeting in April, and they called it the DDD workflow, the discover, distinguish, and designate. And so I don't know if that's what we want to start to talk about, is like, how do you, with the complex autofluorescence that Laura is showing, how do you start to, figure out what you need to do? Well,
that was the third point we're slowly getting at, and I guess it's going to be a segue to start discussing like fun activities to do with your spectral flow data. But your spectral flow is when you have not only high autofluorescence but different signatures that overlap in your same in your sample, essentially. So different type of cells, all of them have different levels of autofluorescence and different channels, different signatures. Everything is kind of mixed together, and now you need to do something with it.
Yeah. So how do we approach that?
Right.
Yeah. So, I mean, the first thing I do is I always just try to do the autofluorescence extraction first. And then if I'm finding unmixing errors, then, and maybe sometimes it's more obvious when you have, what Laura is showing here, or I forget what the other tissue you were showing that has more complex autofluorescence, but,
the lung tissue,
this one. Yeah. So, if I think about it, I think about where is the signature peaking in each of these different examples. And so that's what I would kind of start there, is like, do I have, so like for the first one on the upper right, it's peaking in like, I don't know, V7, let's say, V6 or V7. Then the second one is peaking more in like UV7. And that one's probably a little more similar to the one below it. So maybe it's a brightness level in this case. But if you look at the signature itself, it looks a little bit more similar. And then the last one actually looks pretty much like the first one. It's just dimmer than the first one. But if you look at the actual signature itself, it's pretty similar. So maybe there are only two different signatures in this example, but they each have 2 levels of brightness. And so I would approach it as, let's add 2 different floors, and just take the brightest one to do the unmixing. So sometimes you can see it just by looking at it here. But sometimes it's super complex. If you look at that spectral plot on the left, where, I mean, it just looks like a mess. So, but if you have kind of, because it's a density plot on our spectral plot, and if you have things kind of overlapping and you can see, different areas of peaks and, maybe there are these signatures crossing over each other, that's when I would start to look at the N by N plots of the raw channels. And the N by N plots of the raw channels, I know that sounds very daunting, but we do have a pre-formatted worksheet template. And I'm sure the TASs would be happy to share it with any of our customers. I'm not sure how other companies approach it, but it just allows you to understand, if I plot two, channels against each other, where am I pulling out the most different populations, right? And I haven't really figured out a good way to predict this yet. And I've been trying to think about it and trying to understand, like, what am I looking for? And maybe because from the spectral plot, because it is a density plot, it's hard to understand what you're looking at. But yeah, just looking at those N by N plots of all the red channels, we just kind of go through and see where we see the most populations being pulled apart. And then from there, we can gate on those and kind of look at their spectrums and figure out, are there different signatures, or are they all the same and just different brightness? Where are they peaking? And that will help us to kind of determine what the next step is. So this is kind of like the discover part of that DDD workflow. So
we've been looking at this graph for a bit, and I'm not sure if it's quite clear what's going on. Laura, do you want to explain what we're looking at?
So on the left, I have just the overall signature of this human lung digest. It's just unstained cells. So you can see there's a lot of signal. I think our brightest signal is probably in the 5th decade. And at least in the overall plot, you can, I would say, at least say three distinct signatures visually, but there may be more than that. Then on the right side, I have separated out individual signatures from the overall signature, and Monica kind of discussed the method that I did that. So you basically want to find a single, sometimes you can't even get it done in a single plot, but ideally you're going to start with a single just pseudo color plot where you're going to put two detectors against each other. This is, I thought about putting this as a visual aid, and it was like the one visual aid that I didn't add to this. So you're going to just gate on your cells. You don't need to look at debris, but just gate on your cells. And then I use the N by N plots of the raw data. So the process I use is I just have, basically, if you've used the Aurora and you see the default raw worksheet, which gives you 64 histograms, it's basically that worksheet, but as dot plots or pseudocolor plots instead of histograms. And then I just put any detector on the y-axis, and then the x-axis is all 64 detectors for all 64 plots. And then I look through that and try to find one that has a lot of separation. So if we're looking at this spectral plot, I could actually maybe eyeball it and say somewhere around V9 or V10, somewhere in that region, I'm seeing 3 signatures. So maybe that's one detector that gives a lot of separation. And then I'll actually put that on the y-axis. So I look at V9 versus everybody else. And then I look for the single plot that seems to sort of pull apart all of the different autofluorescent signatures. And you start to see multiple populations, it looks like. And then
draw on the things. I would draw like one signature here, say another one here, and then one on the top here, something like that.
Yeah, something like that. And so then you would get a single plot. So maybe I figured out that V9 versus YG3 for whatever reason. Once I looked at that plot, it actually started looking like I had distinct populations of cells. Then I would gate on those populations, and that's what these gates are over here on the right. So jot these 4 different gates. I actually had more than this, but I couldn't fit them all on the slide. And then once I had gated on the signatures, then I was also curious where those cells landed on the forward versus side scatter plot. So I'm showing that as well. And then you have to do some assessment of figuring out which signatures are actually distinct from each other versus which signatures are the same, but just different levels of brightness.
Mhm.
And then you have to experiment around with putting those into your unmixing algorithm. So even if you find 7 different signatures, then maybe you only use one of the signatures to unmix, or maybe you only use 2 signatures to unmix.
Mhm.
Generally what I've done. But
And so then we have our, and again, if people are using our system, we have a normalization spreadsheet in Excel. And so people can take the MFI of these different signatures in every channel, and then put it in there, and you can normalize to the peak channel and figure out which ones overlay perfectly and which ones don't. And then you know which ones are unique from each other, or which ones are just different brightness level, and once you normalize them to their peak channel, then they'll overlay. So yeah, and we've worked on that. That's kind of part of our process. I think Deanna went through that. Yeah, so she went through, so that's the distinguish portion of the DDD workflow. So the first part is the N by N with the discover, and then distinguish is to try to understand which of these signatures are unique enough from each other that you can use them in an experiment. And then in our unmixing wizard, if you actually put them into your experiment and do the unmixing, it'll show you the normalized signature in the QC controls tab, and it'll return a similarity score. So you can see which ones are too close to other floors that you have in your panel. And that's very helpful to understand, if you should try a different method, or if this is going to be the best way to do it, or if it's going to introduce too much spread because there's too much similarity in the spillover into the other channels. And then, maybe you want to move a floor if it's, spreading in too much into BB510, for example.
yeah, well, I guess there's kind of 2 components to this, right? So there's assessing your unstained tissue so that you can figure out how to unmix it best. And obviously you're going to need to unmix your data in order to analyze your fully stained sample. And so you should, if you have a really complex sample, you're going to have to go through this process to get your data to unmix. Or there's also even a step before that, is doing this kind of assessment before you even design your panel. And based on the assessment of autofluorescence and what the overall signature looks like, then you can make decisions about what fluorophores you should even think about including into your panel for your first panel design.
Absolutely. Yeah, and one of the things I was thinking about as, we were preparing for this session, I think a lot of people don't normally record enough events for their unstained control. And so it needs to be, especially for this, if you have a very complex tissue, you really need to have many more events than you maybe normally would think to. I think, by default, most software programs just record like 10,000 events of the unstained. And I would argue that 50,000, maybe more, depending on, the complexity of the tissue and the rarity of some of those cells that have unique signatures, is going to be important to think about.
Yeah, I've found that 10,000 is definitely not great for this technology.
Exactly.
I guess I don't know a ton of people running like just a homogeneous cell line, but I guess if you had that, maybe you could get by with 10,000.
Oh, absolutely. Yeah.
But yeah, again, it's one of those hard things to define because it really just depends on your tissue and what the autofluorescence looks like and how complex it is. So the
Yep.
more complex it is, then the more total events you had to run. So I think that group that ran the lung digest, I think they were just kind of being very overcautious and figured they might as well just run a bunch. So I think they might have run 200,000 events.
Oh, wow. That's
for
great.
the unstained, which made it pretty easy to assess all of the autofluorescence.
Yeah.
So if you can kind of think of it as your fully stained sample and run a bunch of events, then that's going to help you out. You don't necessarily have to do quite that much, but again, it just depends on what your sample looks like.
Yeah. And another trick that we have in our software that we've used before, just specifically for this reason, is you can merge multiple files together. And so, we'll import the unstained a couple times and then just select those files. So the same FCS file, and just merge them with each other to give us more events to use for this purpose.
So
cool trick.
Yeah. I want to start talking about the weird data sets that we have. But before we move on to that, I thinking about the listener of this podcast, and we promised some level of clarity. So I guess we're going to link to.
It got complex very quickly, didn't it?
So
hard topic.
We're going to link to actual presentations where how to unmix and all these step-by-step on how to do what we're talking about. We're just outlaying like a general idea of what you want to do, and other people have presented more detailed steps. So we're going to link to those in the YouTube sections. But I guess the end question is like, that's going to work, right? Once you've dealt with the unmixing, even if you have like tons of different signatures from different cells, you get decent data, or do you find that it's still somewhat?
No, I mean, as long as people have titrated their antibodies and optimized their staining process, we can get very good data. Yeah, absolutely.
All right.
Yeah, I showed the lung digest data to our faculty director, who had only seen it previously on a Fortessa. And I actually haven't really seen what the data looks like on a Fortessa. She said it was usually just a mess, but I showed her the Aurora data, and she was very impressed with how well it had worked out, even though it's quite a challenge to figure out how to set up the unmixing to work well for you. But if you can get it figured out, you can be pretty successful.
And then you can store those controls, those signatures, right? So you can keep those and then use them for subsequent experiments. Have you found that seems to work for that assay? Although those are human samples, so there's much more variability.
Yeah. We actually don't really tell a lot of people in our facility about the library reference controls.
Okay. I gotcha. Or just reusing them in the experiment.
Yeah. I think some people might reuse them, and that's another video I have. It's explaining when you can reuse controls and when you can't.
When they're good.
Yeah. But yeah, I guess that was the goal for the human samples would be to, spend the time to figure out what is the best way to set up the unmixing for those samples, and then get the controls entered in the reference control library so that they can be utilized for all the patients over time.
Yeah, right.
But that project hasn't really quite gotten to that stage yet.
Yeah, I think it's different with, animal models like mouse, because you have much more, they're genetically more identical because you're usually using one strain. But yeah, with human samples, it gets more complex. And then especially if you have different treatments that you're working with, I'm wanting to say that some chemotherapy agents can cause autofluorescence in cells as well. And so I don't have any evidence of that, but it's more just discussing with some other people. But that can be a challenge. So even the same patient, if you had multiple samples across, a treatment timeline, you could have different autofluorescence characteristics just based off of that. So
Yeah, I think that gets into one of our examples that we were talking about. So we've kind of
Segue. Nice done.
Hey, look at us. That was a nice segue.
So we were thinking we could talk about a few example situations, kind of common general experiments. And with those experiments, the types of things you would need to consider while setting up that experiment. So one of the experiments is this human lung digest that I've been helping out with in our facility. And Monica kind of started talking about it. Let me pull up some example data. So here are 4 different patients, unstained lung digest. So we've kind of started talking about how you mentioned the reference control library, but I guess that's kind of more for the fluorophores. I don't know if anyone ever entered an autofluorescence signature into the reference control library. I've never thought about that before.
I don't think you can because you have to record it directly in there. So the only way you could reuse the autofluorescence signatures would be to just reuse the file, the control file, in the experiment.
Yeah, so I guess the main question that I would think of, which I didn't fully understand before doing this project, is does every patient need their own unstained control? Because looking at these 4 different patients, to me, I think it's very good that we had an unstained control for every individual patient. How this experiment was set up, that just one patient was being run per day instead of running multiple patients per day. But I think it's good that we had that setup that forced us to have an unstained control for every patient. I didn't attempt to use one autofluorescent signature for every single patient. I kind of treated each patient individually and unmixed each patient individually.
That's a lot.
Luckily, it was less than 10 patients total.
That's good. That's good. Yeah, that's a good question. So it's interesting because I'm looking at this and clearly number 1 on the upper left is much more different than the other 3. And then you would argue that the 2 on the right are much more similar to each other. Yeah. And for those people that can't see what we're looking at, we're just looking at different signature, densities, because again, the spectral plot is a density plot, but there's a much more prominent signature in the, let's say, patient 1, where I see some far red emissions that are popping up off of most of the, if not all of the lasers. And I don't know if it's just because there is, maybe that patient has more eosinophils, for example, in their lungs, right? I think eosinophils have more farther red autofluorescence, I believe. So it could be maybe those other signatures are there in that patient. It's just that one's more prominent because of the cell types that are more prominent. So yeah, you kind of start to wonder about, what these different things are telling us.
Yeah, I mean, this is the first time that I personally had worked with human tissue digests. Most of my background is in mouse work. So I don't have a ton of experience with human samples and how much variability comes from patient to patient, but it definitely seems like in this case with the tissue digest, the autofluorescence is quite different in these patients.
Yeah.
I don't know.
And so I'm, and I think of like PBMCs, right? So normal healthy donors, I would think you could use one unstained control for all of them because, I would say usually that autofluorescence signature of lymphocytes, monocytes, they don't change that much just in normal healthy donors. But then if you're getting into taking blood from patients that have been treated with something or they're on, maybe they have more inflammation or something like that, that might be where you would need, even just for blood samples, separate unstained for each patient, or maybe one unstained for each treatment type, for example. But this, and that might be, where I would maybe start to wonder is if there's different treatments or different disease states. But of course, disease states can be variable among humans as well. But then with such a complex tissue, to me, I would say, yeah, I agree with you. I would probably have a separate unstained for each patient.
Yeah, I think it's kind of that, it's similar to people when people ask about, well, should I bring FMOs for every patient? Like you can't answer it until you've run some patients and then you figure out, oh, I should have had an FMO for every patient, or I should have had an unstained control for every patient. So it's probably going back to that argument, then you should run a couple of trial samples and see how things look. And then based on your small trial of a small number of patients, then hopefully you'll figure out if you definitely need an unstained control for every patient, or if you can get by with just one. Or if, depending on what groups you have, if you have treated versus untreated or disease versus healthy, if those two categories have different autofluorescence patterns.
Do you want to talk about the DsRed experiment?
Sure. So we have another situation. This is now a mouse experiment where they have this DsRed fluorescent protein in their mouse. So we were thinking we could generalize this to, any group that's looking at fluorescent proteins in mice, which always adds a complication to your experimental setup. This particular experimental setup is a DsRed cell line, cancer cell line that they're injecting into mice. So they get a DsRed positive tumor. And then they have a immune panel. So actually the immune cells are not anticipated to be DsRed, which is kind of
that makes sense.
Interesting setup to their experiment.
Right. So then are they gating out the tumor cells?
Yes, I think they also have some sample prep stuff as well to clean up the sample. But yes, they would be gating away the DsRed. So their experimental difficulty is that they have mice that have DsRed tumors, and then in order to get their single stain controls, they would need DsRed negative tumors. So that would mean that they need to have a separate mouse that they inject with DsRed negative cancer cells just to get their controls set up.
Is that because the markers are not expressed on like spleen cells? Like they can't just use spleen cells for single stain?
Yeah, some of them. Well, it's also you would want the autofluorescence of the tumor tissue, right?
Only in the unstained control.
But in order to an unstained control that doesn't have DsRed in it, they need
Oh, sorry. When you
DsRed.
said
The
I was
complication.
single stain controls. Yeah. No, that makes sense.
I
Yeah.
guess I lump the unstained into the single stain control.
Yeah, you're right. Yeah. Sorry.
Yeah.
I was
So this
thinking
is
single stain. Yep.
this is an additional layer of complication.
Complexity.
You know,
Yeah.
you might also just have a mouse with a
Uh-huh.
fluorescent protein. So then you would also need to get a mouse without the fluorescent protein and grab that same tissue,
Right.
get the matching autofluorescence of your
Mm,
same tissue that doesn't have a fluorescent protein in it.
mhm. So they so they could take the cell line without the fluorescent protein and inject that as a control.
I think that's the best case scenario.
Yeah, if they have
I
it.
know they haven't been doing that. So I came up with a workaround for them, which is what I'm showing on this slide.
Great.
So this is their DsRed single stain control. And the best workaround I could come up with, even though, not the most perfect situation, but I think it gets the job done, is basically using your DsRed unstained. So you haven't added any fluorophores, but you still have DsRed in it.
Mhm.
Figure out which detector will separate that out. So it's a dim signal. So the main separation is in the first few detectors of the yellow-green laser. So I'm showing YG2 here, and I gated on the most positive signal in the YG2 detector and the most negative signal in the YG2 detector.
That makes sense.
Basically in the software, you can right click on that gate and say, create FCS file from gate or whatever the terminology is. And so you can isolate those two signals. So in the P2 gate on the top, we have what I'm calling the DsRed signature. And then the P3 gate on the bottom is the autofluorescent signature, the DsRed negative signature. So my thought was that they could separate this out and then take the P3 gate and also use that as their unstained control, which could be utilized for autofluorescence extraction if they wanted to take that out.
I think that sounds great.
That's the best.
That makes sense to me. Yeah, absolutely.
If you can't, so you either have to think about, do I need an additional mouse that doesn't have my fluorescent protein so that I can use it for both the autofluorescence assessment as well as my single stained controls? Or can I use this kind of trick and basically manufacture a unstained control out of your fluorescent protein single stained sample?
I call them in silico controls.
But then that also brings up the issue of reference controls and what are the best controls to use for, or what's the best tissue to use for your reference controls. So there's lots of options for your reference controls here, right? So they have tumor tissue for their fully stained sample. So in an ideal scenario, they'd probably want to use tumor tissue for their reference controls if they choose to use cells.
Yeah.
But then we have to think about what is going on with the fluorescent protein. So we need a different mouse that doesn't have the fluorescent protein in it so that we can use that for our controls. But also, I mean, this kind of has an intermediate amount of autofluorescence, I would say. So it might still be okay to use these for reference controls. But I found that when you have, like, for example, the human lung digest with that really extreme case of autofluorescence, it's actually really challenging to use those tissues for reference controls because everything gets so convoluted between the autofluorescence signals and the fluorophore signals. And how do you piece out the fluorophore signal from the autofluorescent signal? At that extreme, it can just make the reference controls much more complicated.
Right.
Then we can start to think about, is it better to use a different tissue that has less autofluorescence for your reference controls?
Yeah, and so I would say, it goes back to like how many cells are you going to get? So to me, like this tumor sample probably won't be very many cells. And so I might already want to use compensation beads for my single stain controls, or like I said, spleen. Like if I'm sacking a mouse, I can take a spleen if I'm not using it for anything else. But you have to consider if those markers are expressed sufficiently and brighter than your sample. And if they are, then you can certainly use spleen. If not, then you have to figure out what's going to work better.
We started this podcast by saying that sometimes autofluorescence can be not just a pain in the neck, but also useful. And I think Monica had something about that.
Oh, so I said we can also talk about when autofluorescence is fun.
Yes.
And I think this goes, well, I mean, if you think about defining autofluorescence in the first place, as Laura, when we gave the introduction, there are compounds in pretty much every cell type that, or structures in the cell that cause autofluorescence. But then there are also, organisms or cell types that have, it's sort of like autofluorescence, but they're actually much more distinct. And for example, chlorophyll has a very distinct signature. And I don't know, like it's autofluorescence because it's internal, but it's not like the typical autofluorescence that we think about. And so I guess it's a little more fun.
It's like the DsRed example we just talked about.
Yeah, it's a little more fun. And so, we've been working with different people from different institutions on various, either cell types or different organisms. We've looked at cyanobacteria. I think the webinar by Alexis Gonzalez goes through some of the marine biology that they've looked at, or marine organisms that they've looked at. But algae, phytoplankton. We've worked with somebody with plant cells. Again, going back to chlorophyll, and then there have been some publications on like label-free cytometry as well, which I guess you could think of it as kind of the same thing. And if you start to look at these organisms or these different cell types, you can actually use high dimensional analysis to separate out different spectra just in the unlabeled cells. During, I think it was the second full spectrum cytometry breakout session at Cyto Virtual the other week, that David Galbraith was talking about, autofluorescence. And I think he called it a fluorotype. So thinking about the internal fluorescence of different cell types, and you can kind of not model it, but start to analyze it using the 64 dimensions of data that we have, and also forward and side scatter too. And you can really start to separate out these different cell types. And of course, he's been working with plants, I believe, throughout his career. So, like I said, he called it a fluorotype because you have different chlorophyll signatures in the plant cells, and you could start to separate those out. I think it was one of the last slides that Deanna presented, and it was an interesting UMAP plot that showed different, I believe, marine organisms or and they, the UMAP was able to kind of put them in different populations. And, I always like looking at these plots. They look like you could turn them into a work of art and put them on the wall. But it's very interesting to see. Actually, I can pop it up. Oh, these are algae that she was looking at. So super interesting that, all these different algae species could be, well, I believe they merged the samples first, but then used UMAP to, kind of separate them out and distinguish them from each other. And you can see how well it did. And like I said, these plots are always so interesting to me because, I look at this and I go, yeah, those shapes look like algae. I don't know. But like I said, it's interesting to kind of start to think about autofluorescence as fun and something we can really look at and analyze and, start to understand about the different cell types or organisms that we're working with.
It's fascinating. At the University of Chicago, we have the DsRed Family Institute that just opened, and it's a massive investment in trying to understand the microbiome world and its impact on human health. And their big chunk of their effort is trying to understand, identify different bacteria and different, and there's not that many tools in flow cytometry to look at the bacteria. So I'd be curious to see if we can manage to use or identify the cells just based on their autofluorescence, trying to tell them apart based on.
Yeah, absolutely.
Yeah.
Using that to distinguish different, and again, label free, or maybe just one or two other labels in addition to autofluorescence.
Right.
Yeah.
Well, that was fascinating. I think I'm gonna stop here. I really enjoyed this discussion. The one thing that I feel is gonna happen because of spectral flow and these dealings with autofluorescence is change the way we work in core facilities. Like typically my entry-level staff will end up on cell sorters. And now that cell sorters have these kind of high-end, pretty complex capacity, spectral flow, autofluorescence dealings, and then some of these machines can now do dimensionality reduction stuff, and then you can gate on those and sort on those, and then it's going to be really complex for the person that you just hired and knows nothing about flow cytometry, and suddenly they need to do. It's going to be a challenge. So really appreciate your time talking to us.
Yeah, it was enjoyable. Thanks so much for the invite. Like I said, it's always fun to talk to you guys.
Oh.
Hopefully we have outlined for everyone listening that autofluorescence is really complicated and you should be thinking about it.
Yes.
Even in conventional flow.
Yeah. All right. Thanks everyone for listening. Again, YouTube is a very lonely place, so leave comments and questions and whatnot. We will talk to you later. Thank you so much.